Our next talk is going to be from Dr. Pedro Fontes. He's an associate professor at the University of Georgia. He's also a beef extension specialist. And he's going to be talking to you guys on some bull development stuff, so the impact of bull overnutrition during development on fertility.
All right. Can you all hear me? Yes? Perfect. Well, thanks, Shelby. I appreciate the opportunity to be here and share a little bit of the work that we've been doing with sire overconditioning. The way that I organized my presentation today, basically over the next 40 to 50 minutes, what I'm going to try to do is basically give you an overview of some of the observational studies that have investigated the potential association between sire overconditioning and fertility in those young bulls. Then I'm going to also share with you some of the more, what we would call experimentally controlled data, where we actually are experimentally getting bulls overconditioned and then evaluated semen quality on those bulls. And then as we move forward towards the end of my presentation, or maybe half of my presentation, I'm going to share with you some of the more recent research that we've been working on. And basically what we're doing, we're not only looking at the impact of overconditioning on semen quality itself, but we're actually taking that a step further. So we're looking how the diet of the bull can potentially influence early embryonic development and fertility from a pregnancy establishment standpoint. All right, so basically my background is predominantly with female reproductive physiology. A lot of my training and also the research and extension program that we do is very female oriented, but you know, since I started in Georgia, it's been about 5 years that I've been at the University of Georgia now. We have a pretty well established program there, which is the Georgia Bull Test, which is basically a consignor based bull development program where producers in the state enroll their bulls and we basically develop those bulls. And at the end of the program, we organize a sale. And, you know, interacting through that program, one of the big concerns that we get from commercial producers is, in fact, that idea that bulls might be getting a little bit overconditioned, right? So I started getting a lot of questions with regards to what extent that could potentially be influencing fertility. And if you walk around the country, that's probably, if you're in the seed stock business, you probably get questions related to that as well. So we were really interested in trying to dig into a little bit and try to understand a little bit more so we can generate some data to back up any potential associations between sire overconditioning and fertility. All right, so to kind of start that, right, when I get questions from producers and I don't know the answer, I call some of my colleagues, and sometimes what we'll do as well, we'll start browsing through data, right? We go in the literature and we try to find studies that might have potentially explored that idea that overconditioned bulls might have altered fertility. So if you start browsing through the literature, there's a lot of good work out of Canada, okay? And this specific work is epidemiological data. This is data out of a veterinary clinic in Canada where they basically reported the percentage of bulls that passed the breeding standards exam based on body condition score. And in this situation here, just want to brought your attention that body condition score there in that figure, it's in a 1 to 5 scale, so not a 1 to 9 scale that we're used to. But basically you can see that whenever we get bulls coming through a veterinary clinic that are a little bit under condition, yeah, we see a decrease in the percentage of bulls that pass the BSC. But also if you go all the way to the right side of this figure here, and this will be your bulls that are going to be overconditioned in a 1 to 5 scale, you can see that the probability of those bulls to pass the breeding standards exam also goes down, right? In this specific situation, like I said, this is data from a veterinary practice, okay? So, and this includes bulls from all different sorts of ages, right? And the reality is in the industry, the bulls that are more prone to get overconditioned are bulls that are being developed, usually in a dry lot setting, right? And getting ready for a sale. And we're able to generate some interesting data that way. So this is data that we generated in our group, okay? Reaching out to veterinarians that were working in 3 different bull development programs, consignor-based bull development programs. And if you look at this figure here, this is basically what we would call a histogram with the distribution of bulls, of our bulls, about 700 bulls in the study, according to different levels of back fat thickness, okay? And this is back fat thickness based on a carcass ultrasonography, okay? So if you look at this distribution of bulls, what we did in this study was, what if we divide that population of bulls in 3 groups, okay? Where we have, here on the right, or, you know, hear me? For the top 10% here on the right for subcutaneous back fat thickness, and then here on the left, we have our bottom 10%, so these will be our leaner bulls. Whereas in the middle here, we have the bulk of the population, which represent 80% of the population of bulls here. So basically, we got these bulls. Those bulls were coming from different operations, and based on the carcass ultrasound that we performed to report to the breed association, we ranked them based on subcutaneous back fat thickness. And then when you look, and again, this is observational data, right? When we look at the percentage here all the way to the left side, this is the percentage of morphologically normal cells in the study, according to the different classifications. Here in the middle, we have the percentage of primary and secondary abnormality when it comes to sperm morphology outcomes of a breeding sinus exam. And what you can see is for each one of these metrics here, the percentage of normal cells was significantly less in the bulls that were in the top 10% of the population for subcutaneous back fat thickness. And this was driven by both differences in primary and secondary abnormalities. So if I bring this figure back here real quick, remember, those are the bulls in the top 10% of the population. So one of the questions that I get is like, well, if there is an association of bulls getting overconditioned, how do I know if my bulls are overconditioned or not, right? So if you look at this figure, the data that I just showed, we were using about a 0.5, 0.6 threshold here to differentiate those bulls in the top 10% of the population. We did look into, okay, so those bulls have a little bit more sperm abnormalities if they're a little bit overconditioned, if they're more on the overconditioned side. But is that enough to influence the percentage of bulls that pass the breeding sinus exam? And here's what I have. This is what I'm showing to you here on the left. So here I have the percentage of bulls that fail the BSC based on the different categories. And you can see that the bulls that are in the top 10% for subcutaneous back fat. And again, this is not the back fat APD, okay? This is phenotypic back fat thickness based on the carcass ultrasound, okay? And you can see that you see a significant increase in the probability of those bulls failing the BSC. And these are failures strictly associated with semen quality. So if there's any other reasons why those bulls failed the BSC, they were not included in this data set. Okay, because again, we're interested on the semen aspect of it. And then here on the right, we have the same data set, but plotted a little bit different. So basically what you're seeing there is the probability of bulls to fail the breeding sinus exam regardless of the category. Here we have based on their subcutaneous back fat thickness. And I apologize that this is in centimeters, but you can see really clear here at 1.2, so that's about a 0.6 inches for subcutaneous back fat. At this stage, we start seeing a really clear increase in the probability of bulls failing the BSC. All right, so if you want to know more about this study, the specific study, there at the University of Georgia, we have a podcast called UJ Biff Tips, and the graduate student that compiled the data, Molly Smith, she recorded a podcast, so you can scan that QR code and you can access that podcast later on and hear a little bit more about that specific study. Then I think the natural question is like, okay, people are concerned about it. When we go there, we look at those observational studies, we can pick up a little bit of a difference there, right? So I think the natural question as a researcher is like, well, can I go there in one of our university herds and generate that phenotype purposefully, right? Can we, in a control environment, get bulls over condition? And if we can accomplish that, do we see differences in semen quality? Because then it's in a control situation. It allows me a little bit more flexibility to understand what's going on. And when you browse through the literature and you go back to Canada again, there's some research out of Canada doing that, and these are bulls that are developed either in a more forage-based type of treatment or in a more high concentrate type of treatment at a John Costellick's group. And basically what they show here is that bulls that were, and again, those bulls were fed these different diets for 170 days. Okay, so that's close to 6 months there. And you can see that the bulls that were in the HG, so this would be your high concentrate, high gain treatment, they had greater scrotal circumference at the end of the feeding period. And then they were evaluated motility and morphology. Those bulls had a slight decrease in motility and a slight increase in morphology defects here. One of the really neat things that they did in the study is that they had these really neat infrared cameras where they measured the temperature of the scrotum. And basically the idea was, and basically what they evaluated or what they reported is, when they evaluated the bulls in the HG treatment, again, those are the bulls that are exposed to the high concentrate diet for, again, about 6 months here, they saw a decrease in the temperature gradient of the scrotum. And let me take a minute to explain that to you. So if you look at this figure here on the right, you can see that, the red part of the image is a little bit warmer, right? And as we go from the top to the bottom of the scrotum, the bull naturally cools that scrotum down. And that's important for the process of sperm production, right? And what they noticed in that study is that the gradient, so the changing temperature from the top to the bottom of the scrotum, was decreasing those overconditioned bulls. And we have done some of that in our studies. We struggle to replicate that, but one of the reasons we think this might happen is, if you look at that figure all the way to the left of the screen, okay, this is a figure of one of our studies where we harvested bulls that were exposed to an overnutrition treatment. And what you're seeing there, so basically here, you can see the two testes as well as the penis of the bull. And right where what we call the spermatic core there, which is a very important part of the reproductive tract of that bull, to cool the testes down, you can see that there's quite a lot of fat accumulation there, right? So one of the hypotheses that we think might be happening is that whenever those bulls get overconditioned, we have a little bit of an impairment on the ability of that bull to control the temperature of the testes, to cool the testes down, okay? Here in the middle, what you're seeing is the vascular structure right on top of the testes, so this is how it looks like anatomically. So basically, the bull anatomically has this whole array of blood vessels here that were developed to cool the testes down. So if we take you back to this figure on the left, that's exactly where all that fat is accumulated. So one of the theories is that those overconditioned bulls have impaired testicular thermoregulation. So here on the right, again, this is a figure from our group. So we have a bull with really good gradient from the top to the bottom of the scrotum when we use an infrared camera. And then when you look at the image on the right, you can see a bull that you have very little thermoregulation of the testes there, okay? But again, this is not something that we observe in every single study that we do with those overconditioned bulls, but that has been reported in the literature as one of the potential mechanisms. So one of the good reasons that we usually think about pushing bulls from an energy standpoint, right? Besides, we're interested in identifying those bulls with genetic potential for growth, right? By no means I am advocating against trying to identify those bulls, okay? But one of the reasons that we normally think about this is the idea of puberty, okay? So the idea that after weaning, we need to push those bulls really hard, otherwise they won't be reaching puberty. And I'll bring a study here, and don't get mad with me because this is in hosting bulls, okay? But it makes sense. I'll show you, it makes sense. The reason why this is done in the hosting bulls is because in hosting bulls, again, we're weaning those calves at birth. So it really allows us to go and control how much energy those animals are consuming during the first 6 months of life. And that's what this study was doing. They basically developed those hosting bulls, and this is out of Ireland. They basically developed those hosting bulls either in a diet that elicited a high body weight gain or a low body weight gain. And then at 6 months, they got these bulls that were developed either in a high or a low plane of nutrition, and then broke them down and two other treatments. So they were either kept in a high plane of nutrition, or they were exposed to a low plane of nutrition. And the same thing happened with the bulls that were in the low treatment. So you had high, high, low, high, and low combinations based on the 6-month threshold here between these two different diets. Those of you that might be in a cooperator program, right? This is something that I like to share with folks that are in a cooperator program, because sometimes you're receiving those bulls from folks that might have, we have gone through their operation and transfer embryos, but if those bulls during early life, they were exposed to a suboptimal condition from a nutrition standpoint, that might impair their ability to really pass a BSE. And I'm going to show you some data that's going to support that. So when you look at the study, here you have the different combinations of plane of nutrition during the first 6 months of life or the second 6 months of life of those bulls. And what you can see here is that age of puberty, okay, was not really influenced by the plane of nutrition in the second half of that study. So this will be considered after weaning, right, in a beef bull population. But the reality is that nutrition during the first 6 months of life was significantly influencing the ability of those bulls to reach puberty. And we see something very similar in heifers, okay? There's a window of developmental plasticity on the brain of replacement heifers. We don't understand a whole lot in bulls, but it seems to be the same case, where if you have a period of suboptimal nutrition during early life, you really hurt the ability of those bulls to reach puberty in time to pass a breeding standards exam. So, and I think this is applicable in a cooperator herd type of situation, or in a situation that you might have suboptimal condition during early life from a nutrition standpoint of those bulls. So, if you start studying overnutrition, I think there's not a mammal in the planet that has more issues with overnutrition than us humans, right? And if you start going through the fertility literature in humans, it's quite interesting because scientists spend a lot of time understanding how male obesity influences fertility in humans, right? And when you look at the literature in humans, we know that males with increase or in an obesity scenario have been shown to have increased scrotal temperature, similar to what has been proposed in bulls. Not only that, they also have some other issues with sperm, oxidative stress, DNA fragmentation in that sperm. But one of the things that really intrigued me is the differences that they see in embryo development. Okay, so this is some data in a human IVF clinic with couples that are struggling to become pregnant, right? And then they go to an IVF clinic, and this is epidemiological data in humans, where they basically got records for those human fertility clinics and sort the records based on body mass index of the male partner, regardless of the body mass index of the female partner. And what they noticed is whenever a couple was getting a treatment for fertility and the male was classified as obese, so that's a body mass index here greater than 28, those couples had on average decreased fertilization rate during an IVF program. But not only that, when they followed those embryos that did fertilize, they noticed that they produced fewer embryos in general that were classified as transferables. Another thing that is really interesting, out of the embryos that they did classify as transferable embryos, when they transfer that embryo back, after producing in vitro that embryo, the probability of that embryo generating a clinical pregnancy was also decreased if the male partner was obese, okay? And if you talk to fertility doctors, very often they make a nutritional intervention on the male if a couple is struggling with fertility issues and the male is overweight. So looking at that and thinking on the data that we're generating in bulls, I think that's our natural next question was, well, does that happen in bulls, right? So if I go there and get bulls over conditioned and a bull goes through and passes the breeding standards exam, do I see issues post-fertilization, right? That out in the field, yeah, we cannot do it, but maybe in a research setting, we can come into the university and test that idea. So We then developed the hypothesis that whenever we get those bulls over-conditioned, we induce a series of metabolic changes in those bulls. Those changes will alter the biology of that sperm cell, and then whenever we go there, that sperm cell fertilize the embryo. That sperm cell might potentially influence the ability of that embryo to successfully develop. One thing that is interesting, and I'm not going to show you here for the sake of time, but sire conception rate. Sire conception rate, which is a metric commonly utilized in the dairy industry, is associated uh, uh, with basically fertility in the field, has some relationship with the fertility of bulls in an IVF setting. Not always, but sometimes bulls that have, some data sets show that bulls that have had really high fertility in a hosting population, in an AI setting, in an artificial insemination setting, they have decreased in vitro embryo production in some data sets. So with all that in mind, we try to test that in overconditioned bulls. And the first study we did was just a preliminary study. And what we did, we got herd bulls. Okay, those are mature bulls that were off season. And then we experimentally fed those mature bulls 2 diets. Either a diet to get bulls over condition, where they had a target average daily gain of 4 pounds a day, okay, or a diet to maintain body weight. So that was our control treatment. And in this situation here, those bulls were fed for a total of 67 days. Okay, so at the end of the feeding period, we went there, collect those bulls, we cryopreserve their semen, and then we ship those to Florida, where our collaborator John Brumfield there performed the IVFs for us in those bulls. And what was pretty neat in that preliminary study, and this is the first study we did with this, we look at cleavage rate, and then we didn't see any differences. And basically, cleavage rate is the proportion of oocytes that actually underwent the process of fertilization. That whenever in an in vitro reproduction system, we dump the sperm in the dish, what percentage of the eggs that were there actually started cleaving, which is an indirect, not perfect, but indirect way that we can see whether fertilization occurred or not. And when we look at those bulls that were gain or the maintenance diet, there were no differences between treatment. When we look at blastocyst rates, and these are basically the embryos that develop all the way to a transferable stage. So for those of you that might be doing IVF at your place, most of the embryos that you're going to be transferring, they're going to be at the blastocyst stage. What we did is we see what percentage of oocytes actually make it all the way to the blastocyst stage. And what we saw is that whenever we used semen from the bulls that were purposefully fed to get over condition, we saw a decrease in embryo production. So those sperm cells were able to fertilize the egg just fine, but whenever we follow those embryos, they're more likely to die before they reach 7 days of age, which is that blastocyst stage. So there's something happening to the sperm that is influencing the ability of those embryos to develop. And again, these bulls passed the breeding standards exam, okay? But these are mature bulls, right? And you're probably thinking with yourselves like, well, but mature bulls, you know, if you go there and feed those bulls, you gain 4 pounds of weight. They're going to gain 4 pounds of fat, right? Those bulls are not growing, very little musculoskeletal growth. So we're basically, yes, inducing a phenotype that is not realistic to what we see in the industry, right? So we didn't try to replicate that in young bulls. So this is an ongoing study that we have where basically we test the same hypothesis in growing bulls, because again, that's the population of bulls, me and you, that's the one we're interested in, because when we're developing those bulls and we're getting those bulls for sale, those are the bulls that are more likely to have a little bit more subcutaneous back fat than we would like them to have. So when you look at this study is quite unique because we use the University of Georgia herds and we actually bred cows, everybody to the same sire, okay? And then we got all these, 44 total, we got all these half-sibling bulls to kind of control for the genetic variation of the sire and randomly assign those bulls to one or two treatments. So half of these bulls were developed were fed for 114 days a moderate gain diet. So those bulls were gaining about 3 pounds, about 2.7, that was the target weight. Target average daily gain, 2.7 pounds per day. Whereas our high gain bulls were fed to gain as much as their genetic potential would allow, but focusing a little target weight was 4 pounds per day. Apologize, this is in kilograms, but the target weight again was 2.7 pounds in the moderate gain group and 4 pounds or greater in the high gain group. And we have a feed intake monitoring system, so we could monitor exactly how much those bulls were being fed, and we could control intake as well in that system, that automated feed bunk system. And the dietary ingredients between the two diets are very similar. We just tweak the proportion of each ingredient around in order to increase the caloric density of the diet of the bulls in a high gain treatment. So um, just to give you a little bit of an idea on how that works. So basically, those bulls are all bunk broken, right? So we brought them into those facilities and they were adapted to the facilities for about 7 days. And then we started the feeding period that, again, like I said, was a total of 114 days. And then throughout the feeding period, there was 4 time points here that we ran those bulls through. We collected body weight, body condition score, and we also did a carcass ultrasound to see how those bulls were changing body weight and carcass composition over time. Okay, and we also went there and collected blood samples as well. And I'm going to show you some of the metabolic markers that we measure in those bulls. The other thing that we did is at the end of the feeding period, we went ahead and performed what we call a serial semen collection. So we run those bulls through, we do a cleanup collection, let them get rid of that old sperm that has been stored in that epididymis, and then we collect those bulls 3 times. Okay, kind of every other day we go there and collect those bulls. And the reason why we don't collect them only 1 time is because from ejaculate to ejaculate, there's some variation there. So we wanted to minimize that. So we went there and collected those young bulls 3 times over the course of a little bit over a week there. And then we performed a series of semen analysis. So we didn't only analyze semen traditionally for motility and morphology, we also used some more modern techniques like computer-assisted sperm analysis, as well as flow cytometry. And I'm going to show you all that data. It's going to make a little bit more sense as we go through. But basically, we got these half-sibling bulls, collected them, prepared morphology slides, and then we immediately extended that semen as if those bulls were in a stud facility. We extend that semen, cool that semen down, adjust the concentration, take it to the lab, and then we do a computer-assisted sperm analysis that we measure all those detailed sperm movement patterns, and we also do a flow cytometry, which you think of, and Dr. Zoka is going to discuss a little bit more on that. You can think of as a little bit more detailed version of a morphology analysis. Okay, so it gives us a little bit more insight on the health of that sperm cell when we utilize a flow cytometry instead of just a morphology analysis. And then when we look at the results of that study, as you can see, this is the performance data, and I'm not going to spend too much time here. I just want to show you that those bulls gain way more than we were expecting, both in the high gain treatment as well as the moderate gain. But still, we were able to generate quite a divergent plane of nutrition on those bulls. And you can see they were gaining about 1.5 pounds difference between the 2 treatments there. What we did as well is compile the data for, on the left, you're seeing body condition scoring, and on the right, this is the subcutaneous back fat on those bulls based on the carcass ultrasound. So you can see from a body condition scoring standpoint that we are able to really get those bulls over condition, right? Which is what we're trying to accomplish. But not only that, we not only subjectively when we look at body condition score, but also objectively when we go there and perform a carcass ultrasound. And like I said, this is in centimeters, okay? But I will convert to inches for you right now. So the bulls in the high gain treatment, they were right about a 0.6 inches in subcutaneous back fat. So if you look back, think back on the first study that I showed you, where the probability of failing the BSE increased right about 0.5, 0.6 inches of back fat, we were able to generate exactly what we were shooting for, right? So we wanted to bring those variations that we see in fertility in the field and then generate treatments that have the same amount of subcutaneous back fat, and we're able to do so. This is exactly the same as our observational data. Some of my colleagues will say, this is not as fat as some bulls that are out there. And I was like, okay, but this is pretty divergent plane of nutrition, okay? So what was neat is when we go there and look at all the blood parameters, and one of the things that we see in humans is insulin dysregulation, right? Obesity in humans leads to insulin dysregulation in what we'll call a type 2 diabetes, right? And that's based on insulin resistance. So these are metrics of insulin resistance. And what I'm showing you here in this figure on the left and the one in the middle is that the bulls that were exposed to the high gain treatment, they have greater insulin resistance when you compare to the moderate treatment bulls. What was interesting, if you go all the way to the right in that graph, that's haptoglobin, okay, which is a systemic marker of inflammation. And you can see that those bulls had an upregulation of that systemic marker of inflammation, which is also something that we see in humans. We get pro-inflammatory markers in circulation, they get upregulated in situations of, I guess we wouldn't call it overcondition. In humans, we would call it obesity, right? In a situation of obesity, you have upregulation of those pro-inflammatory markers. When we go there and actually look at the semen analysis, what was really interesting is that the difference we saw in semen characteristics, they were there, but they were very subtle, okay? So if you look at differences on average for total motility, progressive motility, you can see a tendency for a decrease in the high gain compared to the moderate gain treatment. And the same is true when you look at the percentage of emotile sperm. And again, these are metrics that the computer-assisted sperm analysis gives us, that we don't usually get those, some of those, in a traditional breeding sounds exam. The other thing that I want to point out is that we use a 60% threshold to consider those semen samples that we collected eligible for freezing. So if any of you might be developing bulls and you might take it to a stud facility, they're going to be collecting bulls. In this situation, if we set up a 60% motility threshold for prior to freezing as a threshold for considering that sample eligible for cryopreservation, you can see that the percentage of samples that were eligible for cryopreservation was significantly decreased in the bulls that were exposed to the high gain versus the moderate gain bulls. So that overnutrition model is decreasing the ability of those bulls to meet the criteria for cryopreservation if we were to take those bulls to a semen collection facility that use that criteria of 60% motility, okay? And then when we look at the flow cytometry data, it's quite interesting that we see no differences between the bulls. Well, basically what we did is we evaluated that sperm, and then we incubate that sperm for 30 minutes and we evaluated that again. So we stress that sperm a little bit to see how that sperm reacts to stress. And what you can see there is that right at 30 minutes of in vitro capacitation here, you can think of it as a stressor to that sperm cell. We start picking up differences between the treatments where the high gain bulls had a lesser percentage of sperm cells with intact membrane. So a sperm cell with a disrupted cell membrane is associated, a large percentage of sperm cells with disrupted cell membranes is associated with decreased fertility, and that's what we're seeing in those samples after we incubate them for 30 minutes. Keep in mind that at 0 hours we saw no differences here. And then for acrosome integrity, we saw very minor differences. The only differences that we've seen was a little bit of an increase in the percentage of sperm cell that were classified as having small damage in the acrosome membrane, which is the little hat that the sperm has, that it's important for the process of fertilization as well. And increased acrosome damage is also associated with decreased fertility as well. But now to the nice part, we also perform IVF with those young bulls that were exposed to overnutrition. And what we did is, and as you can see here, this is the percentage of what we call cleavage rate. So out of all the eggs that those sperms were able to, that were eligible for fertilization, when we use semen from those young bulls that were exposed to the overnutrition treatment, we see a decrease in cleavage rate, okay? Meaning that fewer of those sperm cells were able to fertilize the egg and then start the process of embryo development. But then when we follow those embryos, we saw a decrease in the percentage of blastocysts on day 6 as well as on day 7 relative to the number of oocytes and relative to the number of cleaved structures. But it's interesting that we just see the differences on day 6 and day 7. So if you're utilizing these bulls in an in vitro embryo production system here at day 7, that's when your IVF lab will be loading those straws and send it to your place. So you have had a decreased proportion of embryos if you utilize bull semen, based on this study, that was generated um, from bulls that were exposed to overnutrition. We took that a step even further, and we actually we use a technique called immunofluorescence, where basically that's the way we can go there and label the cells of the embryo to allow us to actually count those cells and see how many of those cells are alive, how many of them are dead, how many are going to become the placenta, how many are going to become the embryo. And those cell numbers are associated with the quality of that embryo as well, okay? So here you can see that the embryos that were generated with the bulls exposed to the high gain treatment or overnutrition, they had fewer cells, okay? And they have a decreased number of we call trophectoderm cells. These are the cells that are going to become the placenta, okay? So that was the number of cells in those embryos were decreased if those embryos are generated with semen from bulls that were exposed to overnutrition. Not only that, we use a marker that gives us an idea whether cells are dying or not, undergoing what we call apoptosis, okay? And what we saw is that the embryos that came from the bulls that were exposed to overnutrition, they had a greater percentage of cells classified as undergoing apoptosis. So those embryos not only had fewer cells, but they had a larger proportion of cells that were classified as undergoing cell death, okay? Highlighting and matching with some of our previous studies showing that whenever we expose bulls to overnutrition and we generate embryos, we're not only influencing the quality of the sperm, but we're eliciting some changes post-fertilization as well, which biologically is really interesting. So the last piece of data that I'm going to show you from our IVF outcomes, you know, those guys in Florida that helped us with this, John Bromfield and his graduate student, they do an outstanding job, and they have this machine that has a time lapse during the process of embryo development. And that's what I'm showing you here on the left. These are the percentage of embryos that reach each one of these embryo developmental stages. And you can see that starting early in the first cleavage event, you start seeing a decrease in the percentage of structures whenever you utilize semen in IVF from bulls that are exposed to overnutrition. The other thing that that time-lapse system does is shows us what percentage of cells have cytoplasmic fragmentation, which is also associated with the quality of that embryo. And you can see as early as the 2-cell stage, those embryos are already different if they were generated with semen from a bull that were exposed to overnutrition. So just to summarize, whenever we get bulls over condition and use them in an in vitro embryo production system, we have a decrease in cleavage rate, increase in cell fragmentation, cytoplasmic fragmentation. Those embryos I didn't share the data here, but those embryos take longer to reach the morula stage. When they reach the blastocyst stage, they have fewer cells, and a greater proportion of those cells are dying, okay? But not only that, they take longer to hatch and start growing in vitro, okay? So those embryos grow at a slower rate if they're generated from a bull that has been exposed to our overnutrition treatment here. And again, we're very fortunate to be able to do that in the context of a university setting that we can really control and dial in the diet to generate the phenotype. We are sometimes too stubborn and too curious, so we keep looking at things and trying to characterize the effect of overnutrition. So I got a graduate student working on a project now that we have some preliminary data that I'm going to share with you, where basically we got those embryos that we produced through IVF and transferred back to cows. And we basically synchronized the recipient cows in an industry standard synchronization program, produced those embryos through IVF from bulls that were developed in different planes of nutrition, and then we transfer those embryos to those recipients. And then about day 15, after we had fertilization happening in the IVF lab, we go there and we harvest those embryos and we study how those embryos grow. So if you never saw a day 15 embryo, this is a video of a day 15 cow embryo in a syringe here. You can see that they have this floss looking morphological characteristics. They look just like a floss, like a tooth floss. Okay. And at this stage, this is an important stage of development of the embryo because that's where a lot of the embryos die. Okay, that's where a lot of the pregnancy loss that Rick Funston mentioned in his first presentation. That we beef cows they experience a lot of pregnancy loss, same as dairy cows, and same as virtually all mammals, as humans as well, right? And that stage of embryo elongation is very important for pregnancy establishment. So we wanted to understand if the diet that we're feeding the bull is influencing how well that embryo is growing in that really key stage of embryonic development as well inside the cow. So then, after my student here, my graduate student Lucas, flushed these cows, he's much better than I am at flushing cows, so our recovery rates was good whenever I quit flushing those cows myself and I let him do it. And then what we do is we get those embryos and we cryopreserve them and send them to a laboratory where they do what we call a transcriptome analysis, okay? Which is a fancy word to a technique that tells us how those cells are functioning. The cells of those embryos are functioning. And when we look at our preliminary data, this is how those embryos look like in the dish. Uh, and this is the differences in gene expression. So all that should say is that those embryos, they are, from a cell biology standpoint, behaving a little bit different depending on the diet that we're feeding the bulls. Is that mind-blowing? I don't know about you, but for me, that's mind-blowing. It's amazing the way we treat bulls from a diet standpoint is not only influencing the quality of the sperm, but we're seeing some carryover effects during early embryo development as well. So, but again, this is really preliminary, and just to kind of start summarizing what we learned over the last 3 to 4 years that I've been working with this. Basically, whenever we go there, we get those bulls over conditioned. We increase adiposity. I didn't show you here, but we increase triglycerides in the and we elicit a very similar response to what we see when we get humans in an overconditioned stage with systemic inflammation and also insulin resistance. Once we collect those bulls, we do see a decrease in motility and an increase in morphology defects in the semen. But those decreases, they're very subtle. And a lot of those bulls do, in fact, pass a breeding soundness exam. Are they sterile? Are they not going to generate any pregnancies? No, absolutely not. A lot of them will, okay? But it seems like those pregnancies might be a little bit suboptimal based on everything that we've been learning over this last 3 to 4 years, like I said. Because when we go there and use those semen straws to produce embryos in the lab, we see a decrease in embryo production. Like I showed you, a decrease in embryo quality, increase apoptosis in those embryos. And then when we transfer those embryos back to the cows, it seems like they're functioning a little bit different when they're growing in those key stages of embryonic development there around day 15 of pregnancy. Now, future directions on this, we're going to keep following this a little bit further. And I believe most of us here, Rick mentioned in his initial presentation as well, most of us here, we are familiar with the concept of fetal programming, right? That depending on what we feed cows during gestation, we're influencing the ability of those fetuses to grow, and that's influencing the performance of the progeny, okay? What we're learning based on other species, there's not a lot of data in cattle. There will be in the next couple of years because there's a few groups in the country that are working with that. But what we're learning is that depending on the diet that the bull gets exposed to, that alters sperm biology in a way that influences post-fertilization growth, but not only that, postnatal growth and metabolism. So it has been shown in rodents that if we get males obese, you induce a disruption in the metabolism of the offspring of that male rodent. So if you think about that in the context of the beef industry, where we have some sires that might generate thousands of offspring, in a single breeding season, right? Depending on how we're feeding those bulls, we might be optimizing postnatal performance of the offspring. I'm a little bit of an optimistic. That's how I like to think. I think it's a window of opportunity for us to learn how we can maybe feed those bulls in a way that we can optimize the performance of the offspring, or we might be harming the performance of the offspring. But again, we need to replicate that in cows. We don't know if that's the case. What we do know is if you look in sheep, there's a recent work coming out of North Dakota State where basically what you're seeing there is growth performance of ewe offspring that were generated out of rams that were either fed to gain weight, the positive energy balance, to maintain weight, or to lose weight. So they fed the rams 3 different diets: overcondition, maintaining the condition, or losing condition. And then they followed the performance of the offspring. And what they saw is, if you look at these lines here, this bottom line, those are the ewe lambs that were sire out of a ram that was exposed to the overnutrition diet. So they were about, their average daily gain was about 10% less when you compare to the rams that were sire by other males, depending on the diet that they were being fed. Again, this is in rams. Does that happen in cows? We don't know. Maybe we'll know in a couple of years. But that's the concept of paternal programming of offspring performance. And that's something that hopefully we're going to learn a little bit more over the next 10 to 20 years if we keep working at this. So with that, I just want to take a minute to thank all the graduate students that actually get all the nice work done. And I'm very fortunate to have some pretty good graduate students. They're go-getters and they really make things happen. So I'm very fortunate of that, and I wanted to acknowledge them and also acknowledge all the collaborators that help us with the different research projects that we do. The problems that we try to tackle are too complex to try to do by ourselves, so we really rely on those guys. And also the funding sources. Most of the work that I shared with you is either funded by the USDA or the Georgia Commodity Commission for beef. So I want to make sure we acknowledge them as well. So with that, I'm not sure how I'm doing on time, Shelby, but if we have time, I'll love to entertain any questions.